The modern era of regenerative medicine launched with an intuitive idea. If a patient has damaged tissue, doctors should extract their own mesenchymal stromal cells and inject them into the site of the injury. The expectation was that these cells would engraft, differentiate, and physically replace the missing tissue. This single premise attracted hundreds of millions of dollars in investment. Clinical excitement grew around the belief that the cell itself functioned as the medicine. But research tracking these cells revealed a massive paradox. The 2002 TOMA study found less than 0.5% of cells remained at the treatment site after 4 days. Hofmann's data showed the same rapid washout. Despite this, the tissues were clearly healing. Since the injected cells vanished almost immediately, the actual mechanism of repair had to be something else entirely. In 2006, Massimiliano Gnecchi and his colleagues at Duke University designed a simple experiment to test where the healing power actually resided. They cultured stromal cells in low oxygen conditions to simulate an injury, then collected the liquid the cells grew in. Finally, they used filtration and centrifugation to remove every living cell from that liquid. Gnecchi injected this cell-free liquid into rat hearts following a myocardial infarction. Even without any cells present, the cardiac tissue healed. The infarct size decreased and ventricular function improved. Gnecchi's work demonstrated that the secretome, not the cell, is the active therapeutic agent. The cells function as the production facility for a biological payload. This cell-free liquid, the secretome, is a coordinated regenerative program. It consists of key soluble growth factors like VEG, FGF2, HGF, IGF-1, and TGF-beta. As this model shows, the secretome works in 2 layers. Free-floating soluble factors bind to the surface of the target cell for immediate signaling. Meanwhile, small extracellular vesicles enter the cell itself to provide a sustained response. Once inside, these vesicles release genetic cargo, specifically miRNA, directly into the cytoplasm. This cargo enables the secretome to reprogram cellular behavior from the inside out. This discovery shifted the clinical objective. Success depends on delivering the secretome itself in a high enough concentration to drive tissue repair. While researchers focused on manufacturing these signals in specialized labs, data emerged identifying platelets as the single largest source of extracellular vesicles in healthy human blood. Platelet-derived vesicles carry a therapeutic payload that mirrors what stromal cells produce. They deliver the same growth factors and the identical regulatory miRNAs, miR21, miR223, and miR126, required to manage inflammation and survival. Platelets also offer a physical advantage. While intact platelets remain trapped by tissue barriers, their extracellular vesicles are small enough to pass into lymph, bone marrow, and synovial fluid. This suggests that evolution built a redundant regenerative system. The same secretome the industry has been trying to manufacture is already produced continuously in every patient's own blood. The ExoSmart device is a CE-MDR Class 2A medical device designed to concentrate this biological redundancy at the point of care. It is a single-session technology that functions with any validated upstream centrifuge protocol. Unlike standard protocols, ExoSmart processes all the plasma, combining the platelet-rich and platelet-poor fractions. This ensures the circulating vesicles in the platelet-poor plasma are captured rather than discarded. This is achieved through tangential flow filtration. As liquid flows sideways across a hollow fiber membrane, the movement continuously sweeps the surface to prevent clogging. A 15 kilodalton molecular weight cutoff allows water and waste to escape while retaining the complete secretome. Validated retention metrics show that PDGF-AA concentrates 2.3 times, PDGF-AB-BB 1.8 times, and VEGF 2.4 times. TGF-beta1 remains at parity with plasma. EGF concentrates approximately 3.7 times, as it is protected inside the platelet alpha granules during the process. In 20 to 30 minutes, the device distills 60 milliliters of blood into 6 to 7 milliliters of concentrated autologous secretome, using the patient's own biology to match the intent of manufactured products. This technology occupies a specific niche. Standard PRP lacks characterized vesicle content. Allogenic exosome products lack secretome synergy and exist in a regulatory gray zone. Plant-derived vesicles contain no human growth factors. The safety of this biological platform is well established. A 2025 ESSK analysis of over 76,000 patients found that autologous blood concentrates have a lowest-in-class adverse event rate of 8.7%. Furthermore, dose-response studies demonstrate that higher platelet concentrations lead to improved outcomes. For the ExoSmart device itself, foundational in vitro testing, led by Dr. Laura Mazzucco, showed a statistically significant concentration of PDT-FBB with a p-value of 0.0077, while preserving platelet morphology. Device-specific clinical evidence is growing, including the 2025 Navarro randomized controlled trial published in the journal Regenerative Therapy. We do not claim this technology cures conditions. The honest position is that ExoSmart is a scientifically rational tool. It captures the biological payload evolution designed for repair and is building its evidence on the most established foundation in the field.